an MA in statistics from the University of Michigan. She then went on to do a postdoc at the University of Pennsylvania. Before joining IISc, she was an assistant professor at Azim Premji University. And now she's, as you know, an assistant professor at the Center for Brain Research at IISc Bangalore. Her research is extremely interesting. It's a marriage of human genetics and data science. She's very passionate about exploring genetic variations in human populations. So she raises questions like, what is the reason that such variations exist? Is it about evolution, about population history? And also, what kind of impact do these variations have? So does it impact on disease? And also about whether inheritance has a role to play. So her group addresses these various questions using bioinformatic and statistical approaches. So I'm so happy to have her here. And Shweta, the floor is yours.
Thanks, Kasturi. I'll go ahead and share my screen now.
Sure.
Can you enable?
Yeah. Just done.
Sure. Okay. Are you able to see my screen?
Yes.
Okay, great. Okay, I'll get started then. Thank you for the invitation to speak here. As Kasturi said, my name is Shweta, and I'm a very new assistant professor at the Center for Brain Research, where we're trying to set up a group trying to ask these questions about, if we look at the genetic variation in the human population, what biology can we infer from it? What does this variation tell us? And today I was going to spend some time talking about a particular class of genetic variation that's common, that you commonly see, and what it can inform us about complex traits. This is primarily, this will be a very broad overview of the field as opposed to our research, but feel free to get in touch with me to ask more specific questions. And at any point, if there is something that's not clear, please feel free to put it in the chat, and maybe one of the organizers could interrupt me. Okay, so as a geneticist, I tend to think about genetic variation along these two axes. And I tend to think about allele frequency, which, so is this a common variant? Is this a variant that that is seen amongst 30% of people, 40% of people, or is it a rare mutation, right? So these are the two classes I think about. And in terms of genomic context, what is the function of these variants in the genome? I think about variants as being coding, protein coding, or non-coding. And for those of you who don't work with human genetics, the fraction of coding variation in the human genome is tiny. Out of the billions of base pairs in the genome, only 2% of the variation that we see, or only 2% of the genome is actually protein coding. So depending on what you want to ask, on what your biological question is, you use very different experimental designs to identify the biology underlying each of these four quadrants. So if you're interested in identifying these rare variations in the human genome, you use sequencing, right? So you use next generation sequencing, where you sequence the entire genome, or perhaps if you're only interested in proteins, you only sequence that part of the genome that codes for proteins, which is a much cheaper method called exome sequencing. But what I'm going to focus on today is the other axis, is the other column, which looks at common variation. And here we use a completely different experimental design, which you've probably heard of as something called genome-wide association studies, or as we refer to it, GWAS. And I'm going to spend the next 30-40 minutes or so just talking about what this experimental design involves. Okay, so before we even talk about the design, maybe we spend a little time thinking about what can we study? What kinds of traits or diseases can we study using a GWAS? All of us in our first genetics classes have started out with the Mendelian paradigm of genetics, where one gene causes one disease. So in this example, you have cystic fibrosis, where a patient who has cystic fibrosis, we think of them as having this one variant of the genome that causes CF. However, most diseases or traits that you think about are what are called complex traits, they're not Mendelian. Which means that it's not a single position in the genome or a single gene that immediately influences or immediately causes disease. disease, but your disease state is a combination of the influences of many genes and the environment, right? What this means is that different people may have the same disease or the same trait, but have different causal factors underlying this trait, right? So for example, you and I may have the same height, but the reasons, the biological reasons we have the same height could be very different. So genetically, I think a complex trait, from a genetic perspective might look like this, where another person may have heart attack, coronary artery disease, which is another complex trait. It's not because they have this one variant in one gene that makes you susceptible to a heart attack, but many different genetic risk factors, and the risk factors here are in orange, spread across the genome, all of which collectively give you an increased or decreased risk of a heart attack, right? So that's what a complex diseases is. These are multifactorial. They have no single cause, which means that we have to think about identifying the causes for these in using a slightly different paradigm. So that's what a genome-wide association study tries to do. We try to identify the genetic variation underlying these complex traits. And intuitively, a genome-wide association study is actually quite simple, right? So what you do is, let's say I care about something like height, okay? So I collect a bunch of people who have a range of heights, right? So that's the people here, from very short to very tall. You collect DNA from all of these people, and you genotype each person, right? So for each person, you genotype them at about 1 million. You test if this position. You can hear me and see my video okay, because I'm getting a message saying my internet is unstable. And what you want to see is something like the plot on the right, where what you want to see is if a person carries an A at a particular position, are they likely to have to be shorter than having a T at that position, right? So that's the kind of association we're looking for in these studies. When we talk about what kind of genetic variation can we study in a GWAS, just a quick quiz question for all of you. In case you don't know, just take a guess. How long do you think the human genome is? It's actually 3 billion bases long, right? So really long. What we're looking for is any position in the genome that varies among individuals, and any position that commonly varies. So in which the alternative allele, so the allele not found in the reference genome, if that allele is found at more than 5% of people or 1% of the people, we call it a common variant. And a SNP. So what we're testing are not all positions in the genome, but these SNPs. And we're testing these for association. So ideally, let's say you had an infinite amount of money. You're funded by a lot of grants, and you can test all positions in the genome for association with the disease. But the challenge of that, primarily, is that it's very expensive, right? You imagine sequencing rupees, if not more, and the imagine sequencing 100 samples or 1,000 samples. This would immediately become prohibitively expensive, and therefore geneticists, in the first few years of human genomics, started asking these questions. Instead of having to sequence every single position in the genome, which is expensive, can we select a subset of positions to test? And that's what they tried to do. They used this biological property called linkage. Please stop me if this is not clear, but I assume all of you have come across linkage in your genetics classes before, where we don't inherit each position in the genome independently. We inherit chromosomes from our parents in chunks, right? So an offspring inherits a bit of both of mom's chromosomes and a bit of both of dad's chromosomes because of this process called recombination, and because of which physically close positions in the genome have similar genotypes, right? So let's look at 3 positions on this chromosome, positions A, B, and C. If you know your genotype at position A, you have a sense that it's very likely that you inherit the same chunk of the chromosome at position B, right? Whereas position C is far away, so you don't know. Recombination could have happened, could not have happened. So knowing your genotype at position A does not actually give you that much information about a position that's very far away, like position C. So this property that physically close locations in the genome tend to travel together from generation to generation to generation is called linkage. And in this context is called linkage disequilibrium. So instead of sequencing every single position in the genome, the first in the first few years of human genomics, researchers identified a few million positions on chromosomes, and they are called tag SNPs, which are informative for the region around that SNP or that position, right? So instead of sequencing this entire region, and I hope you can see my mouse here, you only sequence this one position here in black, and with the assumption that because of a linkage disequilibrium or linkage, the region around the SNP carries a lot of information about the region around the SNP, right? So the actual causal variant could be somewhere in this region, and you would pick up some signal of that association by just testing the SNP alone. So by only genotyping these 1 million or so tag SNPs in a GWAS, in effect, you're actually only sequencing 0.03% of the whole genome, which is a fraction of the total cost. And it gives you a lot more information than 1 million actually because of linkage. The way you do that is, so you have cases and controls, you collect your cohort, you collect DNA from each sample, you fragment the DNA to pieces, and because you know that you want to sequence these 1 million positions, we don't use an Illumina sequencer, for example, but you use these microarrays, which are chips, and these chips have oligonucleotide probes on these chips that test for the genotype at these 1 million positions, right? So these are predefined probes based on the 1 million positions that we are trying to test. And depending on your genotype at that particular position, these probes light up with different colors. So if you have a T, then the probe would light up with one color. If you have a C, it would light up with another color, and so on. So the output of this really would be a matrix like this, where what you see. Is there a question? Okay, I'll carry on. So the output of this experiment is a matrix where what you see is for each subject, for each of your cases and controls, you get their genotypes at how many ever SNPs you're testing, right? So in this case, a few million positions of the genome. And then what you do is for each of these 1 million or so SNPs, you test them for association. What you want to see, right, as a biologist, what you would be really excited by is if you see something like this, right, where in your cases, most of your cases seem to have the A allele at this position, whereas most of your controls have a T allele. And if you see something like this, you could say, okay, well, having the T allele really decreases your chances of having type 2 diabetes. So these are the kinds of signals we're really looking for. And that's exactly what you test for. So we run these statistical tests, usually a very simple linear regression or a logistic regression, where we're testing for the association of each position in the genome with the disease, right? And we use a p-value to test for whether the SNP is significantly associated with the disease or not. I haven't mentioned it here, but the actual p-value threshold we use is not 0.05, but we're running 1 million tests. So it leads to this problem called multiple testing in statistics, where just because you're throwing 1 million tests at a problem, just by chance, many of them are going to come up, give you a positive result. Many of them are going to have p-value less than 0.05. So to account for the number of tests we're running, we use a much more stringent p-value threshold of 10 raised to minus 8, which is 0.05 divided by the number of tests we're running. So we actually use a much more stringent p-value threshold. The results of a GWAS are often visualized like this. So this is what's called a Manhattan plot. This is the toy Manhattan plot, where what you see is on the x-axis, each dot on the plot is each of those SNPs that you have tested for association, right? So there's about more than 1 million dots on this plot. They are sorted from left to right on their position on the genome. So here we are only plotting the autosomal SNPs. There's 22 autosomes. The y-axis is a significance, right? So the minus log 10 of the p-value, which means that the higher up you go on the y-axis, the more significant your result is. And the red line here is that threshold for significance, where we are saying anything above this red line is a significant result. Those are the genetic signals that we think are associated with the disease, right? So here we see that this one region of the genome seems to be above the red line, and therefore this is significant. And then we would go and look at it and see what it tells us about the biology. That was a toy plot. This is an actual result from a GWAS for height. This was a paper published in 2019 where let's start with some important parameters, right? So here, N is equal to 360,000. So here in this GWAS, they studied the genetics of 360,000 people and saw the genetic signals associated with height. That's the sample size. Again, they tested more than one. Here is that red line for significance is right at the bottom, right? So that p-value of 5 times 10 raised to minus 8 is over here. It means that all of these signals that you see above that red line are significant. There are many hundreds of regions of the genome that are associated with height. And if you look at that y-axis, it's the values of this y-axis are no longer 5, 10, 15. They're 100, 190, 200, right? So with these sample sizes, in the last few years, what's been happening is that we've been able to run these GWAS on increasingly large sample sizes, and we're able to identify genetic signals at much more stringent levels of statistical significance, right? So what I would take away from this plot is that there is no one gene for height, right? There's thousands of regions in the genome that influence your height, whether you're a little taller than the average or a little shorter than the average. And this is not just true for height, but for most complex traits, as we will see. This is a really useful plot if you're a biologist, where this was again from the latest GWAS on height, where what they plotted was on the x-axis, the minor allele frequency of a particular variant. So again, each dot here represents a significant SNP associated with height. And the y-axis represents the effect size, which is how much does this variant increase or decrease your risk for height? So if it is near 0, it means this variant doesn't really increase your risk for height. If it's really high up, it means having a particular allele, the height increasing allele at this variant, will increase your height by this much, by quite a bit. Whereas if this SNP over here at the bottom means having this particular allele will really decrease your height by quite a bit, right? So what you see is that all of these are significant variants, right? In pink are those variants that are like very, very significant. They are the common variants, right? Anything with an allele frequency more than 10% in the population, they all have fairly low effect sizes, right? And as you seem to go towards the rarer side of the spectrum, where these are variants found in a much smaller fraction of the population, those are the ones that seem to have these large effects on height, right? They seem to be increasing or decreasing height by a larger amount compared to these common variants, even if all of these variants are actually statistically significant. Why is that? Natural selection. So here, again, if we plot variants along, this is a theoretical plot where we plot allele frequency on the x-axis from rare to common, from left to right, and effect on disease. So on the bottom would be very small effect on height. So having this variant will immediately increase your disease risk by 1-fold or 10-fold or 100-fold or 1,000-fold. So GWAS only looks at common variants, right? So on the right side of this square, and you could either identify variants with very small effects or very large effects. What we want to find are these variants with large effects, right? We want to find these variants that are found in 10% of the population. And if you have this variant, you have a 100-fold risk for diabetes. But we don't actually see these at all. Or, well, we rarely ever see this, right? It's very hard to find variants in this quadrant because of natural selection. Because imagine if you have a variant with a very large risk of diabetes, or very large risk for, let's say, schizophrenia or a severe disorder. Please pick up the call. If you have a very large variant with a very large effect, natural selection is very likely to act on this variant and push it towards becoming less and less common, right? So variants in this quadrant are likely to be pushed towards becoming rare. And that's why GWAS usually doesn't identify variants with very large effects of disease, but with variants with medium or small effects. And the variants that are rare, GWAS doesn't even look for them. We're not even equipped to find them right now. But these are the kinds of variants that sequencing studies will try to find. So one of the things that I sort of briefly touched upon, but perhaps did emphasize the importance of, is the sample size, right? What we've been seeing over the last 2 decades or 3 decades of GWAS is that because the genetic signals we are looking for have fairly small effect sizes, right? They have fairly small influences on disease. They require, in order to be able to find them, we need fairly large sample sizes. So currently, the largest ever GWAS for height included 5 million samples, and that's a huge number, right? For lipid levels, the largest GWAS is 1.3 million, and for Alzheimer's, we're already in the 100,000 range. The first few GWAS were run on low-hanging. The first few GWAS were run on very small sample sizes, and right now they would you sort of rely on a little bit of luck to find signals there. They would work if you're sort of, if there's a very low-hanging fruit signal that you could find, and with phenotypes that are relatively closer to biology, right? So phenotypes that have, for example, a metabolite level or a gene expression level, as opposed to something that's way more complex like heart attack, which is influenced not just genetically, but also by environment and so on and so forth. So just as 2 examples, I just wanted to show you 2 of the first, 2 of the GWAS did lead to very successful findings. So this study showed that a common deletion is associated with risk for HIV infection. And this was run with, I think, 2,000 samples, which is a fairly small number by today's standards. And they successfully identified that having this deletion, this fairly common deletion polymorphism, decreases your risk for HIV infection. Another study, again in that first decade of GWAS, found a genetic signal in this gene called BCL11A, which it's actually a very cool story where, again, this was run with a few thousand samples. And they found that this particular genetic signal allows your fetal hemoglobin to be expressed even in adulthood. Normally, for most of us, fetal hemoglobin stops being expressed. And this was one of the studies that has actually allowed, or that has led to clinical trials for people with hemoglobinopathies, this gene actually is in clinical trials right now. So, but just to give you a sense for how GWAS has been progressing in the context of lipids, which is what I worked on for my postdoc, the first lipid GWAS was run in 2008 with 10,000 samples, and they found 18 signals, 18 genetic signals. This was all run in European samples. The second big GWAS on lipids was in 2010. Huge jump in samples, right? From 9,000 European samples went to 100,000 samples, and identified a corresponding risk in genome associated with lipids. The next, again, almost doubling of sample size, and a huge increase in the number of signals, right? So you can see the pattern here. And in 2018, there was one big difference where, for the first time, instead of studying only Europeans, we're beginning to study individuals from around the world, right? So this was the first multi-ancestry GWAS where there were 300,000 individuals and identified 250 lipid-associated signals. The latest lipid GWAS finally was with more than 1 million individuals. This was still very bad on ancestry, but this was one of the first studies which actually had enough of a non-European representation. So one current challenge of a GWAS, really, which I'll briefly touch upon, is that most of these studies have been done primarily on European populations. And as you can see here, even though this study claims to have a multi-ancestry, to be a multi-ancestry study, it's still 85% European, right? And this is the trend that geneticists currently are trying to change. So, okay, so I've just sort of talked to you about what a GWAS is, but I did want to briefly talk to you about what we can gain from these, right? If there are these variants with such small effects, why are we doing these studies? How can we use them? And this goes back to Kasturi's introductory slide, and I'm really glad she brought that up, because what we are learning is that having genetic evidence for a drug target really increases its chance of it being successful in clinical trials by more than twofold, right? So what this plot actually from just a month ago shows us is that these are on the y-axis are drugs that have failed clinical trials at different stages, right? The bottom are drugs that have successfully passed all phases of clinical trials and have been launched. And on the x-axis is what is the probability of these potential drug targets having genetic evidence. And you can see that all drugs that have been launched have a hugely increased probability of having genetic evidence. And what this means is that if we use the results from these genetic studies in that pipeline of clinical trials, where our chances of false positives, our chances of failure during the various phases of a clinical trial are quite lower, and this could help in decreasing the costs of the drug discovery pipeline. From the biological point of view, what GWAS have really underlined is that most traits are highly polygenic, right? We really have, when we're thinking about humans, it's high time we move away from that Mendelian framework, except in the case of these very rare, very severe developmental disorders, right? So most traits are highly polygenic. And we've seen more than 100 genetic variants associated with schizophrenia, more than 1,000 with height, more than 500 with cholesterol. So we're really learning and really appreciating just how polygenic a lot of these traits are. Individual variants have very small effect sizes. So it's not a single variant that is likely to make you tall or short, but really a combination of genetic variants across your genome. And also, a really important point is that most causal variation is non-coding. So most of these genetic signals we're seeing are not in genes, are not in that 2% of the protein coding genome, but 90% of them actually lie in the non-coding genome. So most causal variation is not affecting the function of a protein, but it's affecting how much it's expressed, or how much, or which cell type it's expressed, and so on and so forth. So in order to sort of study that a little further, one common downstream analysis of a GWAS is to overlay the results of a GWAS, so overlay these genetic signals with other kinds of functional genetics data sets, right? So the advantage of working with human genetics is that there's a wealth of publicly available functional genomics data from all human tissues. So here, what researchers commonly do is you take your GWAS signal and you try to ask, are these GWAS signals likely to be found in those regions of the genome that are also expressed, or that are also open, active parts of the genome, as opposed to inactive parts of the genome in different tissues of the body? What do you say if we do that? Can we see if a GWAS signal for lipid levels is more likely to be overlapping with let's say, with active regions in the liver? And researchers have been trying to do that. And what they see is some really interesting biological insights, right? So this is when they do that for a host of different GWAS. As you can see, each of these panels represents a different traits that have been studied. And within each panel, so I'm going to look at schizophrenia here. What you look for is the enrichment of a genetic signal with, let's say, gene expression signal in different tissues. Okay, so here you see that the genetic signal for schizophrenia is most likely to be overlapping with these expression signals or functional signals from these orange tissues, which are from the CNS, the central nervous system. Makes sense. Schizophrenia is a disorder that we associate with the brain, and therefore we expect these genetic signals to be found to be active in the brain. Great. Similarly for rheumatoid arthritis, right? We see that these genetic signals seem to be overlapping with these biological signals that are active in the yellow set of tissues, which is blood or immune tissues. RA is an autoimmune disorder, so it again makes sense. For some diseases or for some phenotypes, we see some really interesting results. So let's look at BMI, which is the second row, the first panel. Here, what you see is the tissue that is most significantly enriched for genetic signals is not maybe adipose, or it's not the digestive system, but it's the brain, it's CNS. So one of the sort of biological insights from GWAS has been that your genetic signals for BMI are actually enriched in biological signals in the central nervous system, and that's been a very cool story to have emerged from GWAS. In our study last year, we did a similar thing for lipid levels alone. Here we try to separate lipids into LDL, which is the bad cholesterol, HDL, which is what you think of as a good cholesterol, right? You can ignore the three panels on the right and just focus on LDL and HDL. And if you look at the bottom panel, which is looking at your genetic signals for LDL, are they enriched in active regions of the genome, in which kind of tissue? You see that LDL is most enriched in the liver, which makes sense. And HDL is actually more strongly enriched in adipose compared to the liver. So what we see is that these are two things that we call cholesterol. If you go to a doctor, they would tell you, yeah, it's obvious. HDL is, metabolized, and adipose, LDL is associated with the liver. So we expect to see these two differences, but it's very cool to see that the genetic evidence also shows up as having differential function in these two tissues. Your genetics are constant across tissues, but these functional signals are tissue specific. And what you see from a GWAS is that these genetic signals point you to the right tissue that are likely to be involved in a particular phenotype. I'm just going to check how we're doing on time, because I would do want there to be enough time for questions. Okay, so we have about 20 minutes, so I'll keep going. What we also wanted to, what we often always want to do from a GWAS is to then ask, okay, so what? How exactly is this variant that you find associated with the disease? How is it acting? How is it acting on your phenotype? One potential way a variant could affect your phenotype is that it affects transcription factor binding, right? So let's say you have a variant here. If you have an A at this variant, your transcription factor can bind there. It can allow gene expression of a nearby gene. Or if you have another allele, if you have the C allele, it blocks the binding of a transcription factor, and therefore it prevents gene expression of a nearby gene. And that's what we try to ask in our GWAS on lipids, where we overlaid our GWAS signals from lipids with transcription factor binding. So another sort of repeating what I said earlier, which is that when you work with human genetics, that's a wealth of publicly available data. So this a publicly available project called the ENCODE project has transcription factor binding data across the genome for a whole bunch of different transcription factors for a whole bunch of different tissues. So we try to ask, are our GWAS signals more likely to be found in those parts of the genome where a certain transcription factor binds? And if that's the case, maybe we can hypothesize that this transcription factor is really centrally important in regulating this particular trait, in regulating lipid levels, so LDL levels. And what we do see is that we saw these 8 transcription factors that were highly significantly enriched in our GWAS signals. Most of these were well known, but this one particular transcription factor popped out called RXRA, which was previously known to be, a GWAS locus. This was a gene associated with cholesterol levels, but now because of these overlaying effects, we could show that it's likely not to affect LDL levels directly, but by acting as a transcription factor, which regulates the expression of a bunch of genes, and the expression of these genes in turn then goes ahead and affects LDL levels. Just zooming out a little bit, right? I've spoken about polygenicity, I've spoken about transcription factors. How does all of that actually play out in the cell? How are all of these different variants affecting a single phenotype? The current paradigm for complex traits is what's called the omnigenic model, like not polygenic, but omnigenic, where a phenotype is affected not by a single gene, but by multiple genes, because these genes act in networks or pathways. And these authors hypothesized or proposed a model in which your phenotype is directly affected by a set of genes called core genes, right? So if you have a mutation in these core genes, they would directly affect the phenotype, have a very strong effect on phenotype. But your phenotypes can also be affected by indirect effects from many more genes in the genome, which can have small effects on these core genes or can have small effects on these intermediate genes. And what we're likely to find from these GWAS studies are these variants that affect any of these hundreds or thousands of genes in these networks. Essentially, small changes that could affect any edge or any arrow in this network could have cascading effects on the phenotype. And these are the kinds of signals we're picking up. So in the context of a transcription factor, you might imagine that a transcription factor is what's called a peripheral gene, where your RXRA could belong somewhere here and have cascading downstream effects on gene expression of a bunch of other genes, all of which in turn lead to collective effects on your lipid levels, right? So something like this, where your RXRA is a peripheral gene, it's a transcription factor which regulates the gene expression of all of these different genes, each of which have been associated with lipid levels. And it's this collective effect of RXRA on these genes that contributes to an increased levels of LDL. Another thing that GWAS can tell us is this connection between rare and common disease, right? So I've been talking about LDL levels in the context of people who don't have severely high levels of LDL, of cholesterol. These are people with a range of variation in cholesterol, but within the what we call the healthy range. But what happens if you look at the genes found in people with very severe cholesterol levels? These are called cholesterolemias, and people and variants that have been identified from GWAS, which are not as severe, right? Which don't lead to as severe phenotypes. What you see is that these two sets of genes tend to converge upon a very similar set of genes, right? So the plot on the right, I apologize, it's not very clear, but the main message here is that the enrichment of genes you identify from these GWAS of lipid levels is strongly enriched for genes that you identify from these rare cases of cholesterolemias, essentially more severe cases of having high cholesterol levels, right? And these are what we call allelic series. In my first slide, I had made this distinction between rare variants and common variants, right? So you think maybe about Mendelian disease versus common disease. These are completely different things going on, but not really. So what I'm trying to get at is that a common variant with a small effect in the same gene will lead to a small increase in LDL levels, whereas a rare variant with a very high effect in the same gene might lead you to have a cholesterolemia, right? A much more severe phenotype. So looking at common disease is also a way to study a more severe rare disease. These are not completely independent. So sort of just going back to that model here, right? Earlier I had shown you this plot for cystic fibrosis, which is a Mendelian disease, where what we had earlier thought of is that you have a patient with cystic fibrosis will have this one causal variant leading them to have the disease or not. But likely what's happening is that this person doesn't just have that one rare variant, but also what's called a polygenic background, right? It can have other common variant modifiers that may have a smaller or a larger effect on disease. If any of you here is a clinician or a clinical geneticist, you know that even in rare diseases, if we really know that this variant in this gene causes the disease, what's been really confusing is this question of penetrance. Not everyone with the same variant will have that disease. Not everyone with this variant may have, let's say, CF. Why is that? One reason that has been proposed is environment, right? Maybe different environmental risk factors mediate this risk. But another one is that the common polygenic background. Maybe the collective effect of all of the common variants that this person carries is also adds or decreases the risk, the degree to which this rare variant can lead to disease. And so now a new sort of paradigm in human genetics research is to combine these two classes of variants, not study them separately, but to identify new ways of studying common and rare variation together. And in fact, the way we're often studying common variation is called a polygenic risk score, or the PRS, which is the combined genetic risk score from all common risk variants, right? So as I said, each common variant alone has a very small effect size, and which might have immediately led you to think, what's the point? Why are we studying these extremely tiny risks when they're not really telling us that much, right? They're not going to tell you if a person's going to have heart attack or not. But if we collectively combine the small risks from each single variant, we could get a single risk score called a PRS, or a polygenic risk score. It's essentially a weighted risk of all the associated variants across the genome. So let's say the way this works is that you run a GWAS. Let's say pick a disease, right? So let's say you pick you study coronary artery disease or heart attack. You run a GWAS, identify all the regions in the genome associated with this disease, and then I want to find out my risk for this particular disease. At all of those associated variants, I look at my genotype, and I essentially add up my risk by saying, do I have the risk allele at this variant? If I do, then I add up, my risk goes up. Do I have the risk allele at this other variant? If I don't, if I have a protective allele, my risk goes down, and so on and so forth. And what you see is that people in the population have this normal distribution of polygenic risk scores. So most of us, in terms of our common variant risk, we fall in this intermediate risk, because we have, like, let's say for heart attack, right? I may have some risk increasing alleles and some risk decreasing alleles. So overall, my overall genetic risk is probably somewhere in between. But there's like 10% of individuals that fall in the very high risk or the very low risk category. And for these people, the knowledge of that common variant risk can help with early intervention. And in fact, this is one of the areas of GWAS which is closest to perhaps being used in clinical practice. This is a plot of PRS on the x-axis from lowest percentile to highest percentile, right? So in the study, a cohort was, a PRS was calculated for a cohort of individuals, and this was without any knowledge of whether they have heart attack or not, right? And then they went back to these individuals and calculated what is the prevalence of CAD in each percentile of individuals. And what you see is really cool, right? Where for the highest percentile, that individuals with the highest genetic risk of heart disease, you see more than 5-fold risk of actual heart attack. So your common variant polygenic score, even though it's derived from multiple small effects, can actually lead to a significantly higher risk when combined. So these PRS are now in the US, they are being slowly applied to in practice, either in clinical practice or in clinical genetics, to sort of see if people have fallen that extremely high risk percentile, because this is a case of a disease where you actually have interventions, right? So your heart attack usually happens, after your 40s, after your 50s, but your genetic risk, you can know your genetic risk when you're born, right? When you're born. So what this can help with is interventions even before your symptoms arise. And that's one of the use cases where genetics for some diseases is proposed to be potentially very helpful. So in fact, I was talking about the connection between rare and complex disease and or rare and common variation. And this plot actually, again, from the last month really shows how these two come together. So in this in this paper, what the authors did was they looked at hypercholesterolemia, which is very, very high cholesterol levels to the point of it being very severe. And they identified these very rare mutations that we know lead to hypercholesterolemia. Okay, so they divided their group of people. Let's just look at they divided their population into people who were carriers of this very rare risk variant and non-carriers. So if you look at the carriers alone, we would expect that all of them would have heart attack, right? They all carry that risk variant, but we don't. We see a variation in the risk of a heart attack. And the reason we do is that seems to be mediated by the polygenic risk, which is what I was talking about, right? It's even if you carry a rare risk variant, your common polygenic risk mediates or modulates how the severity of the disease risk by that single rare mutation. So what you see is that even for a person with a particular mutation, knowing their common polygenic risk can give you some information about what their risk for a particular disease will be. Okay. One challenge of a GWAS right now is something I had alluded to earlier, which is that it is primarily done in European populations. So in this study, this was actually a very crucial study, a landmark study, which took this polygenic risk scores, which were identified from GWAS on European samples, and asked how well do these work in other populations, right? So if you apply the polygenic risk score from a European GWAS on new data sets from a bunch of other populations, how well is the prediction accuracy? So if you apply a PRS applied on Europeans to a European populations, it does great, right? The populations are very similar. But as you start moving away from European populations, these PRSs start performing worse and worse. So a PRS identified from a European population performs really poorly on an African population. With perhaps with relevance to this particular population, to all of us, right? PRSs on Europeans, for some traits, they perform decently on South Asians, but for other traits, they don't do very well, which means that the results we get from these studies on Europeans are not always generalizable to other populations, right? And that's a matter for concern. We cannot identify a genetic risk that we don't trust applies to this particular population. In fact, the study that specifically looked at this question, how do PRSs estimated from Europeans perform on South Asians for a bunch of these different traits? They found that for some traits, there's no significant difference between Europeans and South Asians, right? Where the black and the orange plots are not very different. But for some traits like LDL, And for BMI, for some reason, a PRS that is, or genetic signals identified from Europeans do not do very well on South Asians, which is actually very intriguing because it makes you think that there might be some genetic signals that are unique to the South Asian, in the South Asian context, that have not yet been identified because we don't have these large enough studies on our populations yet. I'm going to maybe just move on because I think we're running short of time, but I'll just sort of end with this slide, which is that there are a lot of opportunities for these kinds of studies in the Indian population because we're incredibly understudied, right? Most of these GWASs I spoke about are in the millions, but if you look at GWAS in India, you know, we're not there yet. The largest GWASs are still in the thousands or maybe the hundreds of thousands. So that's a huge potential for new discoveries, especially in the context of those diseases where we know that Indians have very high prevalences, like type 2 diabetes. We know we have a higher prevalence and we have an earlier age of onset. Heart disease, same problems. So there are these very interesting questions that are completely unsolved. We have a unique environment and unique genetic structure, which means that we will probably find things that have just not been found before. And in fact, I will just end with this paper that came out, I think, this year that showed that looking at a South Indian population identified both common signals that are shared with Europeans, but also a distinct South Asian set of signals that haven't been identified by looking at Europeans alone, right? And these are the kinds of signals that will really be critical if we want to start thinking about personalized medicine in the Indian context. So I will end there, and just want to say that our lab is fairly new. These are the kinds of studies we're thinking about in the Indian context. So we're always looking for students and postdocs, and happy to take any questions.
Thank you, Dr. Shweta. I'm sure all the participants loved your talk. It was very interesting. We have a lot of questions, so I think we'll take one by one. And so if anybody has a question, please put them in the chat box and we will discuss them. Just a minute. Okay, so the first question is asking us, how can we find the list of genes which are above the threshold value?
So when we test a variant, right? So I'll just go back to one of those previous slides. I mean, that's actually a really deep question, and let's go back. Okay. So when we test each variant for association, we get a p-value, right? So each variant, we get the statistical test gives us a p-value, and then we can say, okay, well, if this p-value is less than a certain amount, less than a threshold, we take that as a significant association. What is harder is to go from that variant to a gene, because as I said, most of these variants lie in non-coding parts of the genome. So there's multiple strategies, and this is an unsolved problem, really. Again, sorry, I'm going to try find that slide. Yeah, this. Right, so the causal variant is here, but how do we know what the actual gene mediating that variant is, mediating the effect is? One thing people often do is just find the closest gene, right? So maybe if the variant is here, we could hypothesize that it regulates a gene that's close by, and therefore that gene is the causal gene. But there might be other genes as well, and there are multiple strategies that we use. We try and see, does this variant affect the expression levels of this gene? There's another class of studies that tries to look at that. But yeah, that's a really good question. Please let me know if I answered your question or not. I know I sort of went on a little bit.
Ma'am, actually from Manhattan plot.
Yeah.
From here, how to get the list of SNPs above the threshold?
Because this is already a table, right? You usually get this as a table where you have, for each SNP, you have a p-value associated with it. So you just identify those that are below that threshold.
Okay, ma'am. Thank you.
Yeah.
Okay, thank you. I'll go to the next question. So Sabyasachi is asking if, so she said that you mentioned non-coding genome. What about the dark matter of genome? Can we study them? Like, as far as I know, dark matter is like 98% of the genome.
Yeah, Sabyasachi, I wish we would retire the phrase dark matter of the genome. Because I think I don't know why we started calling it that a few decades ago, and then now we know that most of the non-coding genome is functional, right? We know it does something, we just don't know what it does. So just because it's non-coding does not mean it's not functional. I think that's one thing to keep in mind, right? Identifying the functions of these is a huge sort of challenge and a huge area of focus in human genetics right now, right? Something a lot of people are working on. And I guess, it's important, right? Because we know that so much of our variation between tissues, between individuals, is not because a protein is completely destroyed. It's because of small degrees of variation in how much a protein is expressed. And therefore, I think understanding the functions of this area of the genome is incredibly important. Does that answer your question? Let me know.
Yeah.
All right, thank you. The next question is by Mohini. She's asking, how effective is GWAS as a non-invasive early molecular detection of cancers? She wants it.
We're not there yet, right? I think so I presented the effectiveness of GWAS in CAD as, doing pretty well or doing fairly well at predicting. But as far as I know, no other phenotype has come close to this level, right? It could be because of multiple things, right? Maybe with cancer, there's more, depending on the type of cancers as well. It could be more environmentally influenced. We don't have enough of a sample size to be able to estimate these polygenic risk scores as accurately as we can for CAD. So the effectiveness of this will probably be very different from phenotype to phenotype, right? Even for, just to be completely, completely open, some people this is still an area of debate. Is PRS still going to be actually helpful? Like, if you know family history, how much more information does PRS actually give you? So the answer to whether it can help with early detection of cancer, the answer is up in the air. Right now, we're nowhere close, right? So, but as we perhaps get more and more sample sizes, or as we define our phenotype closer and closer, where we identify better and better genetic signals, perhaps we will get there. But right now, we're not really there yet.
Okay. I'll go to the next question. Debasrija is asking, to what degree of success can we use GWAS for psychiatric disorders to identify gene loci for susceptibility? You mentioned schizophrenia. What about conditions like ADHD that manifest differently in different people?
Yeah, I think so. My interest in GWAS started with psychiatric disorders, so it's something I try and follow, even though I don't work on it. Schizophrenia has been the most successful in terms of finding loci, right? And these are loci that are well replicated, where we know these are true signals. We know these signals exist. So there's two different ways of looking at it, right? I think on one hand, it has been helpful in helping us understand biology. So GWAS associations were one of the first clues that told us that there was an immune component to schizophrenia, right? One of the first schizosynaptic pruning. So GWAS really that were responsible for schizophrenia. It has also told us other things. Like with GWAS, we can also start identifying like similarity in genetic signals across phenotypes. So we know that there is a large degree of a shared component between schizophrenia, bipolar, major depression, right? There's 80% of the genetic signals for these are actually very similar. So these biologically, I think there's a lot we've been finding. When we think about prediction, however, again, we are nowhere close, right? We're nowhere close to really being able to predict whether someone's going to get depression or get schizophrenia or not. That's not a success at all yet. And part of that is, I think, because of a problem that's specific to psychiatric disorders, which is the problem of homogeneity of phenotypes, which is also what is in your question, right? A lot of these GWAS, they rely on large sample sizes. So our phenotypes are often messy, right? You could have the same person or multiple people could be classified as having depression, but you could have depression 1, depression, bipolar depression, unipolar depression, severe depression, essentially it could be a hodgepodge of phenotypes. it helps because it helps you reach those sample sizes, but it also means that it might be harder to find your signals for more specific subtypes of these. And that's another type of study design that we could start doing, which is you start working with your GWAS of smaller sample sizes, but more homogeneous phenotypes, right? And that might help you identify more cleaner signals, with the caveat that they then become less generalizable to other subtypes of depression, let's say. So in these kinds of studies, there's always a trade-off between how clean your phenotype you want to be and how large of a sample size you have. And depending on the question you have, you could go either way. But most studies so far have sort of focused on the let's increase sample size, but at the cost of keeping our phenotype super clean and super specific.
All right. I think Priyanshu has a follow-up in the same context. She's asking about dyslexia in the same context. Yeah.
I don't actually know much about dyslexia. Yeah, unfortunately, I have not been keeping up with the literature on that study, so I will have to get back to you on that. Yeah, I haven't. I don't know what's been going on in that front.
Okay, we'll move on to the next question. Utpal is asking, can we employ GWAS signal on diseases like depression, anxiety, etc.? I mean, with disease that are more associated with environment and early life surroundings.
Yeah, so my PhD was on the genetics of bipolar disorder, and you would be shocked to know that this thing called heritability, right, which tries and asks how much of the similarity in phenotypes between people can be explained by genetic similarity, right? So something trying to get a sense for how much of a genetic signal is there. You'd be surprised to know that bipolar disorder has a heritability of 80% or 60 to 80%, which is really high, which is actually higher than diabetes, let's say. So there is a strong genetic component. The degree to which it can be
Sorry, your voice is breaking a little bit.
Yeah. How about now?
Yeah, that's
Sorry.
okay. I'll go to the next question.
Yeah.
So Archa is asking, as epigenetics is related to cancer, can GWAS help us find the epigenetic changes and reverse the changes so that cancer cell proliferation can be controlled?
I mean, yes and no, in the sense that GWAS is, and it's usually step one. It's never the last step in identifying disease signals. And so, as I said, a lot of these signals are in the non-coding parts of the genome. So if some of these, we know that some of these signals are associated with methylation, DNA methylation, or histone acetylation, or any of those of an epigenetic mark, these could potentially be tested as targets for epigenetic alterations or epigenetic changes in any phenotype, really. So yes, they could help us they could help us identify potential sets of epigenetic mechanisms to test. But there's a long process from there to actually going in and treating or reversing a disease by reversing the epigenetics. So I think that last step is probably going to be very challenging. But yes, definitely it can give you a list of things to test.
Okay. She also has another question saying, can GWAS be used for a plant genomics and help to find a therapeutic drug that can be used against cancer? As we take the different cell lines of cancer, can GWAS help us to identify that this particular plant product can target cancer?
So this particular question, I think, is outside the context of research, right? Like for that, perhaps you need, it is you could run another kind of association study where you're essentially testing if a particular medicine is, helps with a cancer cell line fitness or not. So I think that's a different approach altogether. However, I will say that all of these studies, everything I've spoken about, has come from plant genetics, right? The first association studies, quantitative genetics, came from a set of people who were trying to do plant breeding experiments in agriculture. So pretty much all the theory underlying everything I've said has come from the field of plant genetics. So it's actually very cool, the connection between those. But in answer to your specific question, I think that's a different paradigm that perhaps doesn't come under this class of study.
Okay. Moving on, Anshu has a question saying, can GWAS help help us to create more specific marker for diseases detection, as sometimes common markers fail to identify diseases?
When you say marker, what exactly are you talking about? Like biomarkers?
Anshu, would you like to unmute yourself or put up a follow-up in the chat box? Yeah, she means biomarkers.
It could, right? So again, I mean, you could go in and just like with drug targets, where you can start, or GWAS gives you a list of variants or a list of genes that you think could be could in some way be involved with the disease. So it gives you a very preliminary association. The next thing you could go in and test is, let's say you find a gene that you think is of real interest, like RXRA, the one I showed you. You could go in and test if this gene, the expression levels of this gene are different in cases versus controls, right? And those are the kinds of follow-up experiments you could do. Again, it could point us to those, but I'd just like to say that these biomarkers are in general hard to identify for these complex diseases because they're so complex, right? A single biomarker so far for any of the things I've spoken about have not been identified. Yeah, that's the sort of short answer.
All right. The next one is by Satyendra. He's asking, what are the main causes of bipolar disorder?
What are the main causes of bipolar disorder? I mean, there's genes, there's environment, there's family, there's every disorder is so complex, and the extra challenge is that, right, the main cause for something is going to be different in different people with a disorder, right? So let's say I have a family history. If I have a family history of bipolar disorder, for me, my genetics could play a really important role, right? There is somebody else who doesn't have a family history. Maybe they don't have a strong genetic risk, but they could have a strong environmental risk. Maybe malnutrition, or maybe early microbiome, early environment, early diet, right? So that's the challenge of this, that each person with a particular disorder can have a very different cause. For some, the genetic, it's I think of it as a set of risk factors, right, that collectively add up. So for me, my genetics may be higher contribution compared to my environment. For somebody else, it could be both, a little bit of genetics, a little bit of environmental risk. For others, it could be the opposite, right? I mean, the environmental risk factors are very high, the genetic risk factors are not. So it varies from person to person, and that's part of the challenge of personalized medicine.
All right. He also asks, what is the natural tenement of depression?
Can you elaborate on that a little bit? I don't completely understand the question. I also don't know what tenement means. I apologize.
Satyendra, would you like to send us a follow-up?
Ma'am, what is the treatment of a depression by the natural process?
As far as I know, there is no treatment. I know there are symptomatic treatments and psychotherapy and medication for some people.
Ma'am, I asked, ma'am, if any patient suffering a long time depression and he taking antidepressant medicines, then he suffering also from antidepressant medicines side effects like sexual low potency and libido conditions. What is your treatment, ma'am? He can stop medicines, then he can also feel again anxiety and depression also.
No, I'm so sorry, but I'm not a trained medical professional, so I will not be able to sort of help with that. But I think it is a challenge, and I think a psychiatrist would be the best person. Or if there's, I don't know if there's a psychiatrist or a clinician in the audience, they could help with this. But I'm unaware of how to handle this particular. It's a difficult question, and I'm not equipped to handle it. Sorry.
It's okay.
All right. Moving on, we have Dr. Girish asking us how you could determine the activity of SNP variants in a non-coding region in a specific tissue, say in brain in case of schizophrenia.
Yeah. So what we have, I spoke about these data sets which are genetic, just genotyping people, but an independent set of data sets exist where you take human populations, or you take mice, or you take whatever model organism, and you have gene expression data from each tissue, right? And you measure using something called RNA sequencing how much each gene is expressed in that particular tissue. So, for example, we have a list of genes with their gene expression levels in the brain, with their gene expression levels in the liver, in the blood, and so on and so forth. So what we can see is, do our GWAS signals, are they very close to those genes that are expressed mainly in the brain? Right? If they are, then we can maybe hypothesize that more activity is going on in the brain at these signals compared to other tissues. That's sort of one way of thinking about it. These are the kinds of other data sets. These are called functional genomics that we use to move from association to function. Right? That's the gap that GWAS itself cannot fill, and we need other kinds of, lots of other data to try and fill in these gaps.
Thank you, madam. Thank you, madam.
Okay. So Abhinanda asks, what is the average cost for a GWAS study of 5K sample size, and who provides the funding?
The second one is a hard to answer question, but so GWAS for a single sample costs about 5,000 rupees, right? So then you can do the math. The funding, I mean, a lot of these studies, right, they are disease specific. So you can apply to either government organizations or to disease or to private funders or non-profits that support research on specific diseases of interest. So these are the two avenues for funding for such studies primarily. But as I said, there's not many going on in India right now. So it's still a fairly nascent field. Right now, one of the sort of pioneer studies, not GWAS, but a population-based genetic study in India is called Genome India, where we're trying to sequence 10,000 individuals from this country. These are healthy people, right? So, but for this 10,000 people, we are collecting some basic traits like red blood cell count, white blood cell count, lipid levels. So hopefully you'll be able to do a basic GWAS on these 10,000 people in the country. But this is just a beginning step. I think there's a lot more to be done.
Okay, thank you. She also has another question asking, for drug testing, which is more relevant, GWAS or biological markers like blood and fluids?
I would say both, right? I mean, the best with biology, any single piece of evidence is limited, and you might get false positives or false negatives for a host of reasons. So I think the targets that are the most likely to be successful are those which have what's called a convergence of evidence, right? Evidence from multiple different sources. So your ideal candidate would be one that has evidence from gene expression, from biomarkers, from genetic signals, from, I don't know, being positive signals from other diseases, so on and so forth. So to specifically answer your question, which is a stronger, which helps more, I don't know the answer off the top of my head. But I think what currently pharmaceuticals are really trying to do is streamline those targets that have as many sources of supporting evidence as possible. And that includes both biomarkers and genetic evidence.
All right, thank you. So we will end the Q&A session at 12:30. If anybody has other questions, we will send a follow-up email, and you could also contact Dr. Shweta. But let us end with the last few questions. So Debangshi asks how GWAS can be used for analyzing metagenomic studies.
So Devanshi, I'm not an expert, but I might imagine that you could, let's say you have a metagenomic study on a group of 10,000 people, right? And you have composition of your phenotype here. A GWAS is essentially an association between your genotype and a phenotype, And that phenotype can be anything. So in this case, your phenotype could be microbiome composition, a composition of a particular species of interest. And you could run a GWAS on, I don't know, Bacillus. Let's say you pick your favorite bacterial species. I'm unfortunately not a microbiologist, but pick your favorite species of interest and use a phenotype as species composition and run an association on that. So those are the kinds of studies you could potentially run. I'm not aware of whether there's many of these being done right now, because I think metagenomic studies, microbiome studies are still, at this point, primarily focused on identifying species composition and things like that. So genetic studies on that are still fairly new, but I think there's a lot to be found there. So these are potentially one way of asking these questions. Like host, essentially asking if host genetics has an influence to play on microbiome composition, right, on your gut microbiome.
Okay, thank you. So next question is by Eric. He asks us, how does ethnic diversity impact studies with large sample sizes when searching for disease-causing variants? And he also asks, what is the impact of genetic diversity on variant classification and its clinical relevance?
So I think of these two as two different things in terms of ethnic diversity versus sample sizes. The primary sort of, there's two reasons it's important to have genetic, or it's important to think about diversity in genetics, right? One is, let me go back to that slide which I skipped over. Sorry. I don't know. Sorry. Yeah, this one. So one of the reasons your genetic diversity is important is that let's say these are two populations, Europeans and Indians, right? We are not, in a GWAS, we're not testing all regions of the genome. We're only testing these tag SNPs. But that the implicit assumption of a tag SNP is that this SNP is in linkage with this region of the genome. This region of the genome tends to travel together in this population. But different populations blocks, because of which, for some of these tag SNPs, you might see that in an Indian population, this SNP is no longer in linkage with this SNP, with a variant here. And what if the causal variant is actually here? It means that you would get a false positive signal in an Indian population if you just applied the results of a European population, right? So these are some of the reasons genetic diversity is important. Another is that these are complex traits where environment is important, right? You can imagine that when it comes to, let's say, something like type 2 diabetes, your environmental factors are going to be very different between Europeans and South Asians. Your diet is different. Childhood nutrition is different. So all of these lead to potentially different gene-environment interactions, genetic signals, and that's why it's important to consider that the same signal that you find in the European population may not hold up. I don't know if I answered that specific question, but happy to talk more in detail offline, Eric.
Okay, thank you. So last question is from Anshu, and she asks, from where can we access the GWAS data for a particular disease?
Yeah, so there are really good resources available for these GWAS datasets. There's most GWAS right now are done on European samples, I said. And one of the reasons for that is that there's a resource called the UK Biobank, which the UK has a centralized health system, which means that you have the genomic data and the phenotypic data for hundreds of thousands of individuals, actually millions of individuals, which can be analyzed together, right? So this has been a huge boon for geneticists. And these GWAS have been performed on UK Biobank extensively. So if you just look for UK Biobank GWAS results, you should be able to find them easily. I would also look up, I'm just going to type it in the chat. There's a lab called Benjamin Neal's lab at Harvard, or at the Broad Institute, which has released these GWAS results for a host of different traits from the UK Biobank. So if you just look up Neal Lab UK Biobank summary statistics or GWAS statistics, you should be able to find it. And feel free to email me and I can send you a link if that's easier.
All right. Thank you so much for this engaging talk and answering all our participants' questions. I'm sure they enjoyed it. So thank you everyone for being here on a Saturday morning. Thanks for your participation. I have pasted some links in the chat box. Please do fill out the feedback form. We will upload the the recording of this session on YouTube. And if you haven't subscribed to our mailing list yet, please do subscribe. And once again, thank you, Dr. Shweta.
Thank you. Thanks for having me.
Okay, I will end the meeting then. Thanks, everyone.